Synthetic Biology, a Primer, by Paul S. Fremont, and Richard I. Kidney is a comprehensive guide to one of the most exciting and rapidly advancing fields in modern science. Synthetic biology is an interdisciplinary field that applies engineering principles to biology, allowing scientists to design and construct new biological parts, systems, and even entire organisms with specific functions. Unlike traditional genetic engineering, which involves modifying existing genes, synthetic biology takes a more systematic and modular approach, assembling biological components in a predictable and standardized way. The book introduces readers to this fascinating discipline by explaining its core principles, historical development, and wide-ranging applications in medicine, agriculture, energy, and environmental science. It is structured in a way that gradually builds the reader's understanding, making it suitable for students, researchers, and professionals who want a clear, yet technically rigorous introduction to synthetic biology. The book begins by laying down the foundational concepts of synthetic biology, explaining how it differs from traditional genetic engineering and biotechnology. One of the key ideas is the use of standardized biological parts, similar to components, in electrical or mechanical engineering. Just as engineers design machines using interchangeable parts, synthetic biologists create biological systems using modular DNA sequences, proteins, and genetic circuits. The concept of biobricks, standardized DNA sequences that can be combined to create functional biological circuits, is introduced as an essential tool for designing synthetic biological systems. Another critical concept is hierarchical design, which involves assembling small biological components into larger, more complex systems in a step-by-step manner. The book also discusses bioinformatics and computational modeling, showing how software tools help predict how synthetic biological systems will behave before they are built. This is a crucial aspect of synthetic biology, as it allows researchers to design efficient and reliable biological systems without excessive trial and error. One of the most exciting areas of synthetic biology is the creation of genetic circuits, which function like electrical circuits but operate inside living cells. Just as electronic circuits use transistors and logic gates to process information, synthetic biologists design genetic circuits using DNA, RNA, and proteins to control cellular behavior. The book explains how biological logic gates, such as AND, OR, and NOT gates, can be used to program cells to perform specific tasks. For example, engineered bacteria can be designed to detect environmental toxins and respond by producing a visible color change, acting as a biosensor. The authors also explore feedback loops and regulatory networks, which allow cells to self-regulate and maintain stability in changing environments. Another important topic covered in this section is CRISPR-Cas9 genome editing, a revolutionary technology that enables precise modifications to DNA sequences. CRISPR has transformed synthetic biology by allowing scientists to edit genes with unprecedented accuracy, making it easier to develop new medical therapies and engineered organisms for various applications. The medical applications of synthetic biology are among the most promising and impactful areas covered in the book. One of the most well-known applications is the development of biosynthetic drugs, including synthetic insulin and antibiotics. By engineering bacteria or yeast to produce essential medicines, synthetic biology has revolutionized pharmaceutical production, making it more efficient and cost-effective. The book also explores gene therapy and cell therapy, where synthetic biology is used to modify human cells to treat diseases. A notable example is CAR T-cell therapy, which involves genetically modifying a patient's immune cells to target and destroy cancer cells. Another breakthrough discussed is the role of synthetic biology in vaccine development, particularly in the creation of mRNA vaccines, such as those used for COVID-19. These vaccines use synthetic genetic material to instruct human cells to produce a harmless viral protein, triggering an immune response. The book highlights how synthetic biology continues to drive medical innovation, offering new possibilities for treating genetic disorders, infectious diseases, and even regenerative medicine.
Beyond medicine, synthetic biology is making significant contributions to agriculture and food science. The book explains how genetically modified crops are being engineered for improved yield, pest resistance, and enhanced nutritional content. Unlike traditional genetic modification, which involves inserting single genes into plants, synthetic biology allows for more precise and complex modifications, enabling scientists to design crops that require fewer pesticides and fertilizers. Another exciting development is lab-grown meat and plant-based alternatives, which are being developed using synthetic biology techniques. By engineering cells to grow in controlled environments, researchers can produce cultured meat, which has the potential to reduce the environmental impact of livestock farming while providing a sustainable source of protein. The book also explores how synthetic biology is being used to develop drought-resistant crops, as well as methods for reducing reliance on chemical fertilizers by engineering plants with synthetic nitrogen fixation pathways. These innovations have the potential to revolutionize agriculture, making food production more sustainable and resilient in the face of climate change.
Another crucial area covered in the book is the role of synthetic biology in the development of biofuels and renewable energy sources. Fossil fuels are a major contributor to climate change, and scientists are exploring alternative ways to produce sustainable fuels using engineered microorganisms. The book explains how algae and bacteria are being engineered to produce biofuels such as ethanol, butanol, and biodiesel. These biofuels can be used as cleaner alternatives to gasoline and diesel, reducing carbon emissions and dependence on fossil fuels. Another innovative approach discussed is synthetic photosynthesis, where researchers are designing biological systems that mimic the way plants convert sunlight into energy. The book also introduces the concept of microbial fuel cells, which use engineered bacteria to generate electricity. These bioenergy solutions hold great promise for creating a more sustainable and environmentally friendly energy sector. While synthetic biology offers incredible potential, it also raises important ethical, safety, and regulatory challenges, which the book addresses in detail. One of the key concerns is biosafety, ensuring that genetically modified organisms, GMOs, do not pose risks to human health or the environment. The book discusses containment strategies and risk assessment methods to prevent unintended consequences. Another major issue is biosecurity, as synthetic biology could potentially be misused to create harmful biological agents. The authors emphasize the importance of international regulations and responsible research practices to prevent such risks. Additionally, the book explores ethical debates surrounding artificial life and genetic modification, including questions about whether scientists should have the power to redesign life at a fundamental level. Public perception and regulatory frameworks are also discussed, highlighting the need for transparent communication between scientists, policymakers, and the general public to ensure that synthetic biology is developed in a responsible and ethical manner. The final chapter of the book looks toward the future of synthetic biology, discussing emerging technologies and potential breakthroughs. One exciting possibility is the development of artificial life, where researchers aim to create minimal, genome organisms that can be programmed for specific functions. The book also explores the role of artificial intelligence, AI, in synthetic biology, as AI-powered tools are increasingly being used to design complex biological systems more efficiently. Another fascinating area is the integration of nanotechnology and synthetic biology, which could lead to the creation of bioengineered nanomachines for medical and industrial applications. The book even touches on the potential for synthetic biology to support space exploration, such as engineering microorganisms to produce oxygen and food on Mars. These futuristic applications highlight the limitless potential of synthetic biology and its role in shaping the future of science and technology. Overall, Synthetic Biology, a Primer, is an engaging and informative book that provides a thorough introduction to the field. It effectively explains the fundamental concepts, real-world applications, and ethical considerations of synthetic biology in a way that is both accessible and technically detailed. The book makes it clear that synthetic biology is not just about modifying existing organisms, but about designing entirely new biological systems with transformative potential. Whether in medicine, agriculture, energy, or environmental science, synthetic biology is poised to revolutionize multiple industries, making it one of the most important scientific fields of the 21st century. The book serves as a valuable resource for anyone interested in understanding how biology can be engineered to solve some of the world's most pressing challenges and create a more sustainable and innovative future.